So we have continuing with the next part of molecular basis of inheritance, that is the genetic code. Now if you look at the nitrogenous bases, we have these 4 nitrogenous bases. So the number of nitrogenous bases are 4. The number of amino acids to be encoded is 20. So if we consider a coding pattern where only one nitrogenous base is used at a time, so the number of amino acids to 1, that means if we say A codes for 1 amino acids, U codes for another amino acid, G codes for another amino acid, and C codes for another amino acid. If we consider this system, then only 4 amino acids will be encoded or coded. If we consider, this is called singlet. You have to remember this term. This is called singlet. If we consider 2, we call it as doublet. In that case, only 16 amino acids will be coded. But if we consider 3 nitrogenous bases at a time, and this system, we call it as a triplet type of system. So what will happen? The number of amino acids encoded will be far more than the required 20. What is the advantage? In this case, the advantage is all the 20 amino acids will be coded. So therefore, it was universally accepted that triplet code, that is 3 nitrogenous bases, they code for 1 amino acid, which was given by Gamow in 1954, and this was referred as a codon. If we consider 3 nitrogenous bases at a time, for example, if we have such type of some system of nitrogenous bases, so this will be 1 codon, this will be second codon, etc. So the universally accepted system of coding, we call it as a triplet code, or we call it as a codon. Now it was Crick in 1961 using mutations, that is, he changed these bases, nitrogenous bases, and he gave evidence that it was the triplet code which was working. The next important phase of the discovery came from Nirenberg and Mattei. And what they did was, in the laboratory, they could synthesize an artificial mRNA. That is, they could synthesize the mRNA by using these known bases. The artificial mRNA in this case is known. And they used this uracil basis. We call it as a homopolymer because all these bases were uracil. In that case, it was found that this sequence, it gave a type of sequence which all the amino acids in this case were phenylalanine. Now what did this indicate? That this UUU or this codon, this codes for 1 phenylalanine. Second, it codes for another phenylalanine. So again, it was proved that it is the triplet code which is working. It was Hargobind Khurana who artificially again synthesized messenger RNA, and in this case he used some different sequences, and he received a Nobel Prize for this. Hargobind Khurana of Indian code. He used this sequence, that is CUC, UCU. Again, he repeated this CUC and UCU. Such type of sequences were used by him. And the type of amino acid sequence synthesized was, this is called leucine, this is serine, leucine, serine, etc. So what does it indicate? That this CUC codon, it codes CU, it codes acid called serine, and this continues. So this was the contribution made by Hargobind Khurana, where in the laboratory he could literally make the sequence of amino acid by similarly a polypeptide chain of such repeats like CUA was also done, and it gave rise to a type of amino acid which was similar. It was also established that there is an enzyme called polynucleotide. There is an enzyme called polynucleotide phosphorylase. This was responsible for polymerizing the RNA. That is, the extension of this RNA happened or occurred in presence of an enzyme, and that was also given by Popov. So this is the basic understanding of how the genetic code came into existence. An important contribution was done by Beadle and Totham. Now Beadle and Totham in 1941, they exposed the spores of fungus, and it was Neurospora crassa. So this is the fungus. Normally they ask this in the NEET examination, so please take care of this. So they'll ask you whether they exposed the radiation to either bacteria or which fungi, etc. So you have to remember that it is Neurospora crassa. To a mutagenic agent like X-ray. And what they found was when these spores, neurosporal, when these spores, they were exposed to X-ray. So if this is X-ray and if these are the spores, and later on they found that these spores which were exposed to X-rays, they were found to be mutant strains. What do you mean by mutant strains? That means if we look at two spores, if we consider this as a normal spore and if we consider this as a mutant spore, so I can call this as a normal spore and I can call this as a mutant spore. That means if normal spore was exposed to X-rays, mutant spores. Now in that case, it was found that this mutant spore, it was lacking some enzymes. So some enzymes were lost. The enzymes, all the enzymes, for example, 5 enzymes were present in this case. Now after that, this mutant type of spore, it contains only 4 type of enzymes. This is just an example to make you understand. So now that was because of certain mutation taking place at the level of DNA, and it was this DNA which was responsible for the formation of an enzyme. In an indirect way, this DNA was responsible for the formation of enzyme. So if there is any change, if there is any mutation in this DNA, then there is definitely going to be any change or loss of this enzyme production. So based on this, it was hypothesized that one gene gives one enzyme hypothesis. So the meaning of this is a part of a DNA which is called a gene. Now if this is a gene, so if this is first gene, if this is second gene, so this gene is going to give rise to one enzyme. This gene is going to give rise to second enzyme. So one gene gave rise to one enzyme. So that was the hypothesis which was given by the initial experiments of Biddle and Totter. One gene and one enzyme hypothesis. Now later it was Ingram who modified this as one gene, one protein or one polypeptide hypothesis. Now the meaning of this is this gene, it will give rise to an, not always an enzyme, it will give rise to something called as a protein, or it will give rise to something called as polypeptide. So all the polypeptides may not necessarily be enzymes. Few or very less number of polypeptides may be enzymes. So therefore, this concept or hypothesis was modified by Vernon Ingram as one gene, one protein hypothesis, or one gene, one polypeptide hypothesis. So that is the meaning of one gene, one enzyme, which was modified as one gene, one protein, or one polypeptide hypothesis. And the reason, as I told you, because many genes coded for proteins which were not enzymes. So not all the proteins were enzymes. So therefore, this modification was done. Coming to a genetic code, by definition, it is the sequential arrangement of bases in the DNA. So if this is the DNA, so these are the nitrogenous bases which are present on the DNA, which controls the sequences of amino acids in a protein. So if this is a DNA, now this DNA, it is transcribed into messenger RNA so that these bases will be copied here, and this, it gives rise to a protein. So that this codon, it codes for one amino acid, next codon, it codes for another amino acid, so it goes on. So by definition, a sequential arrangement of bases in the DNA molecule that controls the sequence of amino acid. So these bases which transcribed into mRNA, then which control the formation of amino acid specifically, or a protein chain in general. So what are the characteristic features of genetic code? Genetic code is always a triplet code. That means it is the three nitrogenous bases, as you can see here, they code for one amino acid. So three nitrogenous bases, when they code for one amino acid, we call them as a codon, or we call it as a triplet code. The second characteristic feature of genetic code is it is universal in nature. What do you mean by universal in nature? Now, if you look at this code, if you look at UUU as a code, this codes for phenylalanine. So this is the codon, we can say, and this is the amino acids or amino acids to which it codes. Now, it is observed that this codon, which codes for a specific amino acid codon, it is present in plants, it is present in animals, it is present in bacteria, it is present in all the microorganisms. So that indicates that the genetic code is universal in nature. That is, the codons code for same amino acids, in all the living organisms. The third characteristic feature is these triplet codes or codons, they are non-overlapping. If you look at this nitrogenous sequence of nitrogenous base, so we have AUG, CCG, AAA. So three nitrogenous bases, they code for the first codon. So this is the first codon. Then the second CCG, it codes for the second codon. Then AAC, it codes for the third codon. Now what do you mean by non-overlapping? Non-overlapping means one nitrogenous base. So if this is a nitrogenous base, this cannot be a part of two codons. So AUG, if it is one codon, then GCC cannot be a second codon. So what do you mean by non-overlapping? Nitrogenous bases of one codon cannot become a part of adjacent codon. So if this happens, so this is a wrong way of coding and this is the right way of coding. This feature we call it as non-overlapping. Now more than one codon, it codes for a single amino acid. We know that the total number of codons are 64. Why 64? Because it is 4 cube, 4 nitrogenous bases and 3 at a time. So they code for 64. There are 64 codons. So out of that, 3 of them we call them as non-code, that is they do not code for any amino acid. Those are terminator codons. So 61, the number of amino acids to be coded are 20 and the number of codons are 61. So therefore more than one codon will code for a single amino acid. So single amino acid will have more than one codons. So for example, serine, this is the amino acid and this will be coded by these 1, 2, 3 and 4 codons. What are those codons? UCU is first codon, UCC is second codon, UCA is third codon, UCG is fourth codon. By more than one codons. We call this as degenerate, or it is also referred as redundant or synonymous. Please remember this. This is normally usually asked in the NEET examination. There are very exceptional cases like methionine and tryptophan which have only a single codon. This is also a most important thing which you have to remember. The next important characteristic feature is these are commaless. Now what do I mean by commaless? Again, if you look at the nitrogenous base sequence, it is AUG, first codon. So this is the codon. Then we have CCC, the second codon. Then we have GCU, the third codon. Now, this has to be continuous, meaning AUG, if it codes for one codon, you cannot, this nitrogenous base cannot be skipped. There cannot be any pause, there cannot be any break in the continuity. So AUG is 1, CCC is 2, GC cannot break. We cannot say CCG is second. So this coding system will be wrong. So what does that mean? It is continuous. The message on mRNA is continuous without any breaks or without any pause. That characteristic feature we call it as commaless. Non-ambiguous. Particular codon always codes for same amino acid. So if you take AUG, it will always code for methionine. If we take GAA, it will always code for glutamic acid. So non-ambiguous means what? It is 100% sure that this codon, it codes for this amino acid. So that characteristic feature is referred to as non-ambiguous. Initiator codon, if you look at the process of protein synthesis, this is the ribosome which is attached, the smaller subunit, larger subunit, and the first, the transfer RNA, it comes with its own amino acid. So the first codon, the initiation of protein synthesis, it takes place in presence of the first codon, and that first codon universally it is AUG, and that is called initiator codon. Initiator means the protein synthesis initiates with the help of this codon, and AUG, it always codes for methionine. In very rare cases, it is GUG, which is also or which also acts as initiator codon. Now, as the protein synthesis takes place, there is this ribosome goes on moving here fixed sequences of codons like UAA, UAG, and UGA. Now, these any one of these at a time, this we call it as a terminator codon. Terminator codon is also called stop codon. Now, stop codon means this is a signal. This codon is a signal that the protein synthesis should stop. So there is no addition of amino acid when the ribosome arrives at this position. So terminator codon or stop codon is something where the protein synthesis will have to stop and the polypeptide chain has to be released into the cytoplasm. Next is the principle of collinearity. The linear order of, as I told you, DNA, if this is a DNA and if these are the nitrogenous bases, this gives rise to this DNA, it transcribes, it gives rise to RNA. And this RNA, it gives rise to amino acids. So the linear order of this DNA, nucleotides in this DNA, it determines the linear order of RNA, and this in turn codons will determine the linear order of proteins, that is the linear order of amino acids. So that is referred to as principle of collinearity. So this is one of the most important topics where many number of questions they appear in the examination. So you have to remember this, and we'll also solve some of these questions which appear from this topic or which have been previously have appeared in different competitive examinations. Thank you very much.